Hey everyone, we're now going to work our way through a series of topics that I have to admit I've really been looking forward to this semester because we're finally at a point where we have all the intellectual tools that we need to talk about and understand some modern biotechnology technologies, and that's going to lead us into some discussion about genomics as well. And part of what this lets us do is it's going to let us make sense of a lot of things having to do with the coronavirus. So I'm going to break this up into a series of 6 videos to kind of hopefully make the videos a little bit shorter and easier to work through. And here's what we're going to cover. So this video is mostly an overview video, a bit about why we're going to be talking about the topics we're talking about. From there, we're going to talk specifically about a series of tools that are used to work with and study DNA. And then after that, we're going to transition to talking about studies of gene expression, so more having to do with RNA and proteins. And then we're going to wrap up this series talking a bit about cloning whole organisms and also about the use of stem cells in biology research. Okay, but for this video, we'll focus on more of the overview. Okay, so let's just take a step back and evaluate where we've come from. We started this section of the course talking about Mendel's discovery of the concept of genes, where each individual has 2 heritable units and things are inherited in a simple dominant recessive fashion. From there, we transitioned forward to Morgan and Bridges' discovery that genes are found on chromosomes. I also talked a little bit about Griffith's study of bacterial transformation, figuring out that DNA is heritable material. Also, Hershey and Chase's experiments contributed to that discovery. Then we talked about how Watson, Crick, and Franklin worked out the structure of DNA as a 2-stranded double helix molecule, and that provided us with some insights into how DNA replicates. And then we saw Beadle and Tatum's experiments on connections between genes and enzymes, and then expanded from there to our contemporary understanding of how genes are expressed. And of course, on top of the people that are highlighted here, there were many, many other scientists involved in these discoveries through time. So the era we're going to talk about now dates from the 1980s or so, or a little bit before that, through this year, so the year 2020. And so we're going to actually start out with this character on the left, Frederick Sanger, who won the Nobel Prize in the 1980s for his discovery of methods to sequence DNA. And we'll also wind up talking about 2 of the 2020 Nobel Prize winners, Emmanuelle Charpentier and Jennifer Doudna, who discovered a technique that lets us edit genes. And just as another historical note, 2020 is actually the first time that a Nobel Prize has ever been awarded to 2 women only. And out of the over 950 Nobel laureates, only 58 women have won Nobel. So this is a pretty substantial landmark in science at the moment. Okay, so what do we need to know about biotechnology? Well, the idea here is to harness what we know about how biological molecules work to start to be able to ask and answer a wide range of questions in biology. And that includes a combination of causal questions and also descriptive questions. So questions such as, so how and why do genomes vary within an individual, within a species, and between species? You know, of a lot of interest right now, how do we know who has a coronavirus and who had it sometime in the past? In addition, how do specific genes contribute to specific outcomes? So for example, what actually causes diabetes, and what causes cancer? For more basic biological questions, so not driven by direct application, what causes things like the crickets I study to have long wings instead of short wings? And why is an organism like a hummingbird so good at flying? And then on top of that, these tools start to allow us to address some of the emergent properties that we've talked about throughout the course. So how is it that different cellular processes interact and give rise to outcomes at other biological scales? And also just a very basic question in a lot of cases of, well, what does thing X do? What does gene X do? How do we know? How can we figure it out? And so again, by the end of this set of videos, my goal is that you will know more about the main method that's used to diagnose active COVID cases, and you should be able to explain that method to other people as well. Okay, so this is kind of our introduction, and now we're going to dive in and talk about those specific types of genetic tools. And just one other note here for the purposes of our course. What I want you to know is just generally how these tools work, and then also know the major pros and cons that I'm going to highlight as I go through and talk about these types of tools. And with that, let's get started.